Worldwide, researchers and industry leaders are improving health outcomes for patients by utilizing the multifunctional efficacy of regulatory T cells to develop novel therapies and disease treatments. Regulatory T cells, commonly referred to as T-regs, play a critical role in maintaining order in the human immune system by regulating and suppressing excessive inflammation. Given the therapeutic potential of T-regs for patients with autoimmune diseases, cancer, and organ transplants, both researchers and industry leaders are developing novel therapies that utilize the multifunctional efficacy of T-reg cells in disease treatments to improve health outcomes for patients. A scientific leader in T-reg function and clinical applications, Dr. Tang is the professor and director at the Transplantation Research Lab in the Department of Surgery at the University of California, San Francisco. The Tang Lab focuses on translating knowledge on the mechanisms of immune intolerance into novel therapeutics for treating autoimmune disease, diabetes, and preventing transplant rejection. Currently, two major areas of her work are focused on the therapeutic application of regulatory T-cell therapy in type 1 diabetes and transplant rejection, or GVHD. In this presentation, she shares the latest research conducted by her lab using T-reg cells for therapy, the benefits of these therapies, and explores a technical challenge of commercializing T-reg therapies that require new approaches in manufacturing and regulatory review. A copy of her presentation will be available for download on the Invitria website.
So why do we work on regulatory T cells? And then it's because if you don't have these cells, and actually it's a human disease, and these young boys actually can have fatal autoimmune diseases, and then that tells us regulatory T cells is critical for immune homeostasis. And then they are really the master regulator of immune homeostasis. And then it's because of their quality list in blue on the left side, is that they have dominance over other cells, and then they also can adapt to different kind of a condition. And then they persist a long time. They actually, once the inflammation is controlled, they also have the ability to promote tissue repair. So this kind of a multifaceted therapeutic effect is really difficult to recapitulate using a single drug or a combination of drugs. That's why we think that regulatory T cells themselves can be a therapy. And then first we need a protocol to grow these cells. And so this is what we call the generation 1.0 polyclonal T-regs. And our manufacturing work stream is slightly different from most of other people have used for clinical manufacturing. We start with blood product, either whole blood or leukapheresis, and then we actually purify regulatory T cells using FACS. And we use three markers, CD4, CD25, and CD127. And then this will allow us to maximally isolate the regulatory T cells with highest degree of precision and purity to enter into the manufacturing stream. And our expansion protocol depends on the use of anti-CD3, anti-CD28 coupled beads. And we give 2 rounds of stimulation, 9 days apart, and then feeding with IO2 and fresh media in between. And then with this, you will see that we get, on average, about 500-fold expansion in 2 weeks, in 14 days. Okay, and I want to emphasize actually the FACS purification we use, which is critical, I think, to our success. As you can see, that most of the T-reg we produce remain to be regulatory T cells at the end that are high in FOXP3 and have a demethylated FOXP3 enhancer region, is because of high purity of the cells we use. If you don't purify the cells, the conventional T cells and non-regulatory T cells actually grow much faster than regulatory T cells, and they take over the culture. So with this particular protocol, the polyclonal expansion protocol, we have embarked on or are embarking on 5 different patient populations and 6 different clinical trials in type 1 diabetes, cutaneous lupus, pemphigus, and kidney transplantation and islet transplantation. Okay, here's some data that's published already that first of all, we found that if you expand regulatory T cells in vitro using this protocol, you actually can repair some of the regulatory defect. One of the criticism or concerns with this technology is if you want to treat autoimmune disease using regulatory T cells, and you isolate these autologous T-regs from the patients, their T-regs may be defective, so that you won't be able to, the cells will not have the therapeutic function you're looking for. But at least for type 1 diabetes, we know that's true, in that if you look at the control, I don't know, do I have a ability to point? Can you see my point? So if you look at the control here, this is looking at IL-2 signaling, phospho-STAT 5, and then if you give IL-2, the control Tregs have high level of STAT 5 phosphorylation and IL-2 signaling, whereas the type 1 diabetes has low level of signaling compared to the controls. After expansion, they're at the same level. And so the in vitro expansion corrected this defect, and then the cells are robustly maintained FOXP3 Treg phenotype with high CD25, high CTLA4, and a high LAP expression, and then they're more suppressive, right? The black bars can suppress as you dilute them out, whereas the gray bars before expansion, they lose their suppressive function sooner. So these cells after infusion can be tracked using deuterium. This is because during the ex vivo expansion, our media contained deuterated glucose. That get incorporated inside the cell into the genomic DNA actually. And then we can isolate the genomic DNA from peripheral blood cells and then measure the deuterium. This allows us to do two things. So one is to follow the Tregs and to see when do they peak or how long do they last, specifically the infused Tregs. Another thing it can do, as I'm going to show you on the right side of the slide, is that it will allow us to track the stability of the cells to see if they change into other cell types. So at least for the kinetics, what we observed is that the cells peaked in the first 2 weeks after infusion, but then there's a very dramatic drop from the peak in the first 3 months. After 3 months, they stabilized and then stayed, and then can be detected as late as 1 year, as long as 1 year. The last follow-up we did was a patient, was 1 year after infusion. And then in all patients, we could detect the regulatory T cells. So they tell us two things. And then first of all, the cells actually lasted much longer than we actually expected. But we wonder why it has this sharp drop from 2 weeks to 3 months. And then whether it's redistribution of the regulatory T cells or some regulatory T cell died, and then they cannot be, they did not persist. So that remains to be investigated. And so on this side of the slide, I'm showing you the stability of these cells. So what we can do here is we can sort the regulatory T cells, or we can sort the non-regulatory T cells and then partition them into different subsets and ask where does deuterium reside? As you can see here, this is different colors, different time, and all the deuterium that can be detected are in the Treg compartment and then not in any of the other compartment throughout the 1 year period. So this suggests to us that regulatory T cell we infuse remain to be regulatory T cells, and then they're phenotypically and then stable. What about the diabetes disease itself? And then in this particular trial, we enrolled 4 cohorts that received 5 million, 5 million, and then 40 million, 320 million, and 2.6 billion Tregs. It's 3 to 4 patients per group, so it's not aimed to establish any efficacy. But from this cohort, what we can see is actually C-peptide, is a measurement of a beta cell function in these patients, remained fairly stable during the entire trial period. So we also use this protocol, these polyclonal T-regs, to treat kidney transplant patient for this particular patient population. And then we have designed a trial to pre-select patients who have inflammation in their biopsy. And then these patients, kidney transplant patient at UCSF routinely get a 6 months biopsy to assess their graft function and graft infiltration. About 20% of patients will have these, what we call the subclinical inflammation. Their serum creatinine is normal, but their biopsy don't look normal. And then this gives us an opportunity to treat the patient population with experimental therapies. And then in this particular case, it's a polyclonal T-regs, because there's no established guideline how to treat this subclinical inflammation. So our intent is actually to conduct a big trial of polyclonal regulatory T-cell therapy. But before we did that, we want to determine the feasibility of this approach. Since these patients are received a kidney transplantation or on immunosuppression, are usually much older and then the type 1 diabetes we have experience with. So we enrolled 3 patients. Here's their age and their cause of kidney disease, and also their enrollment biopsy. All have some degree of inflammation as indicated by these numbers here. They all received about 320 million regulatory T cells, polyclonal regulatory T cells. And the first question we asked is if these cells would persist as we have observed in type 1 diabetes patient, because then again, these patients are on immunosuppression. And particularly calcineurin inhibitor we know are detrimental for regulatory T cells. And so the colored lines here are from these kidney transplant patients, and then the blue lines are the type 1 diabetes patient who received the same dose of regulatory T cells, 320 million. And as you can see, the pattern here is almost superimposable. So we are delighted to see this result, and that at least at this time point, 6 months after kidney transplantation on that dose of immunosuppression, and the regulatory T cells we manufactured can persist in this patient as in the non-immunosuppressed type 1 diabetes patients. And moreover, we did not see destabilization as shown on this side. Again, here the black dots are T-regs and the open symbols are subsets of T conventional cells. And then we don't see deuterium signal among T conventional cells and only see them in the regulatory T cells. So that's reassuring. In terms of their inflammation, because of this trial design, and then we can go back and rebiopsy the patient and actually have a quick readout of the efficacy. And quite like the skin story I'm telling you, that allow us to have some quick biomarker of potential efficacy or safety. So here's our 3 subjects enrolled, and then the first subject was our best subject. Repeat biopsy 2 weeks after infusion, and the inflammation was nearly completely gone. And then this reduction in inflammation was observed at 6 months after the T-reg infusion. The second subject also showed reduction, but more gradual reduction of inflammatory infiltrate. The blue numbers, by the way, are the CD45 positive cells per biopsy. The third subject did not respond, actually showed increase in the inflammatory cell load. We can also actually monitor the effect in the urine, because the patient only have this one transplant kidney producing urine, and all the metabolites and then potential inflammatory markers actually can drain into the urine. And if you measure urine chemokine, which is a marker of rejection and inflammation, and then they were high before T-reg infusion and universally reduced after the T-reg infusion. So it suggests that the T-regs, infusing T-regs in people with inflammation that did not cause exacerbation of the disease, and then potentially it may have some effect in reducing inflammation. So that's our experience with the polyclonal Tregs. From preclinical models, we know that antigen-specific Tregs has much higher potency against organ-specific diseases, be it the transplant or islet in type 1 diabetes. So we then developed this manufacturing process to manufacture donor alloantigen-specific Tregs for transplant indications. So this process starts the same way of using FACS to purify regulatory T cells, but instead of using bead as a primary stimulation, we use donor-derived activated B cells. And then it's very much like a mixed lymphocyte reaction. You mix these activated B cells together with purified Tregs and then feed with IL-2. And then this is about 11 days, the cells will expand and come to a rest. And then we give another round of stimulation with anti-CD3, anti-CD28 beads and to increase their number. And the end product, I'm going to show you in the next slide, are donor and alloantigen specific, but they're polyclonal, as depicted here. They are enriched for alloantigen or highly purified for alloantigen specific T cells, but polyclonal in nature. And then here's the expansion of these donor alloantigen specific cells. This is our actually qualification runs when before the submission of our drug master file. In reality, when we actually entered into patient populations, this become highly, highly variable. In terms of the quality or identity of the Tregs after expansion, they remain to be demethylated at the FOXP3 enhancer region, so that's good. They remain to be Tregs. Here's their suppressive, donor-specific suppressor activity. And as you dilute out these regulatory T cells, and they remain to be highly suppressive, if you use polyclonal Tregs, and they quickly lose their activity. And then same is true here, that if you stimulate it with donor versus third party, suggesting these cells are indeed very specific for donor and highly potent at suppressing donor-specific response. This is at 1 to over 100 dilution of the regulatory T cells. So with this product, we have, we're conducting 4 different clinical trials, 2 in kidney and 2 in liver. And then these trials are ongoing, so I'm not going to go into these trial details for this talk. So the challenges with this platform, and there are many of them, and I circled some of them here. For example, the FACS purification is really our bottleneck. We cannot process more than 1 unit of blood. And then this is already using 2 FACS sorters and then sorting for, 10, 12 hours back, two sorters sorting simultaneously. And then really is not only the limit of our team to work nonstop for about 20 hours, but this, I think, is also limit of the cells, because as you're sorting, they're sitting at room temperature, and then that's also stressful for the cells. So another challenge is actually the use of the beads. And we do 2 rounds of stimulation, so we got to have the precise count of the cells and adding the right number of bead to cell ratio. And then we also have to count during the re-stimulation to make sure we have the right ratio to re-stimulate. At the end of the expansion, these beads need to be removed. It's actually very labor intensive. So our biggest day of the expansion is actually the day 0, and then day of harvest, and then day of re-stimulation. So if we can actually get rid of these beads, it will make our life a whole lot easier. And then the challenge, as I alluded to in the last slide, is this high variability in expansion. So one of the approach we have taken is really a serendipitous discovery by a postdoctoral fellow in the lab. It's actually we end up developing a bead-free protocol that without the bead and then without the need of restimulation, it's quite simple. You give a bead-free cocktail at the beginning of the expansion. So here's the expansion characteristics of multiple donors and then bead-free expansion versus the bead stimulation. And then the bead stimulation here is only one stimulation because the bead-free protocol is only one stimulation. The one thing I think it doesn't really show up very well because this is on the log scale is that the bead stimulation actually take off much faster than the bead-free protocol. And then the bead-free protocol is slow to start, but they're more persistent. And then in fact, our early experience, we stopped the experiment at day 9. That's only, the cells are only halfway done or less than halfway done their expansion. They peaked actually day 16, 17 after the initial stimulation. And this is a more dramatic demonstration of the effect of the bead protocol on day 9. You can see the cells are all come to a rest, whereas the bead-free protocol are still going. And then very strongly aggregated. And then, so we think this bead-free protocol may be a much better alternative for automated production and more robust and reliable production of polyclonal T-regs. All right. So with that, I'd like to summarize and then just to say that we have really the regulatory T cell therapy start to take off, and then they are. We show that this is feasible, and then they're, I think, collectively in the field, more than 100 patients. I think more than 200 patients have been infused with regulatory T cells. We have infused more than 50 here at UCSF. And then they're well tolerated and then seems to be rather safe and harmless. And then the cells can persist for a long time. And then there may be some hint of a favorable biomarkers after regulatory T cell infusion. So the challenge, I think, in this is in regulatory T cell therapy is how do we design trials to read out efficacy and to see if this is a technology and then therapeutic platform worth pursuing. But here on this side, I list other challenges and mostly related to manufacturing since we're going to work on this together. One is this highly variable expansion that especially in disease patient that we want to target. And this sharp drop of infused regulatory T cells on day in the first 3 months is interesting. I wonder if it has something related to the biology of the regulatory T cell we expand. We feed them, we stimulate them very strongly in vitro, and then perhaps that may impair their persistence in vivo. And the manufacturing workflow is extremely labor intensive and extremely expensive, and then it really needs to be changed and need to be automated to make it a therapeutic platform that can be sustainable. And what I did not touch upon, and something else we're struggling, is that this cryopreservation impairs the raw material and then possibly impair the product itself. So that's another area we need to develop.